Bismillahirrahmanirrahim. Assalamu alaikum dear students. Welcome to your channel The Anatomy Canvas. Today in Embryology Foundation Module 3 we are going to discuss the chromosomal structural abnormalities. So chromosomal abnormalities might be defined as changes resulting in visible alteration of the chromosomes. Most chromosomal aberrations are produced by disrepair of the broken chromosome, or by improper recombination, or by malsegregation of the chromosomes during mitosis or meiosis. So chromosomal abnormality mostly fall into two categories: the structural chromosomal abnormalities and the numerical chromosomal abnormalities. So chromosomal abnormalities can be either the constitutional abnormalities, means it may be present in all the cells of the body, or it can be the somatic chromosomal abnormalities that is present only certain cells or tissues. So constitutional chromosomal abnormalities usually occur when there are the structural defects very early in the meiosis or mitosis, and somatic occurs at a later stages, as a result of which the chromosomal are present in very early development and affecting nearly all the body systems, while the somatic abnormalities, some of the body systems are normal and other may be affected, and the affected individual shows a mosaic pattern. So structural abnormalities can be of different types. They can be because of the deletions of a part of a gene or a part of the segment of the chromosome. There may be translocations, there may be inversions or duplication. So we will discuss all of these abnormalities one by one. In this slide you can see this is a chromosome with the short arm and the long arm, and different genes in this area are designated as A, B, C, D, E, F, G, H. So in this chromosome you can appreciate that this segment D is deleted and there is no segment D over here. So the affected individual will show the genes which will be present over here. So the gene defects will be present in such an affected individual. So these deletions can be small or they can be large. They can be affecting the mutations in one gene or affecting many genes. There may be homozygosity, that both the allelic forms are affected, or there may be heterozygosity for each. And the consequences of the deletions can result how much the gene functions are disturbed, and this leads to different effects in the individuals. So deletions leads to, because of the loss of the chromosomal material, they can be large scale deletions, and large scale deletions may be lethal. So example of the deletions are the Cry-Du-Chat syndrome. In Cry-Du-Chat syndrome there is a deletion on the short arm of the chromosome 5 which affects motor and mental functions. Infant cry resembles a mewing cat. Special chromosomal breakpoints are associated with specific phenotypic changes. In this chromosome, you can see this is a chromosome number 5. This is the centromeric position. This is Q arm and P arm, and in P arm this is the locus 15, 1, 2, 3, where the genes for the larynx development and gene for the nervous system development are located. So when there is a deletion in this segment, it leads to Cry-Du-Chat syndrome, and in Cry-Du-Chat syndrome there is variation in phenotype associated with the region deleted, and the researchers have identified regions with gene involved in larynx and nervous system development. So Cry-Du-Chat syndrome is partial deletion on short arm of the chromosome 5. The clinical features are micrognathia, malocclusion, the cleft lip and palate, cat-like or mewing cry, the microcephaly, the round face, low set ears, and also the epicanthal folds, the hypoplastic nasal bridge. There may be hypotonia, there may be hypertelorism, and there will be the mental retardation. So these are the different features which are associated with Cry-Du-Chat syndrome. And Cry-Du-Chat syndrome, again I repeat, it is the deletion on short arm of chromosome number 5 in the area which are responsible for formation of the larynx and the nervous system. Angelman and the Prader-Willi syndrome. So deletion on chromosome 15 causes both these two types of the syndrome, and this is actually a pattern which shows the genomic imprinting, because if the chromosome 15, which is paternally derived, is affected, it can lead to the Prader-Willi syndrome, and if chromosome number 15, which is maternally derived, it is affected, there are deletion on it, it will lead to the Angelman syndrome. So if from the father, the Prader-Willi syndrome is characterized by obesity, low muscle tone, cognitive disabilities, and incomplete development. And if from the mother, Angelman syndrome, severe mental retardation, developmental problems, speech problems, problems in movement, unusual facial appearance, apparent happy moods, prolonged and unprovoked periods of the laughter. There may be muscular abnormalities and the hand flapping, etc. So these two different diseases, the same chromosome, the same gene are affected, but the origin depends upon whether it is maternally derived or it is paternally derived. So the gene from the father is affected, lead to Prader-Willi syndrome, and if the gene from the mother is affected, it can lead to the Angelman syndrome. So these are the different chromosomal pattern in Cry-Duchet syndrome and in Angelman syndrome. And in this you can appreciate this is the chromosome number 5 where short arm is deleted, and these are the areas where the chromosome number 15 is affected, leading to either Prader-Willi syndrome or the Angelman syndrome. So structural abnormalities can also be because of the translocations, and translocations means exchange of the chromosomal segment between the non-homologous chromosomes. So there may be the two major types of the translocation. This can be the reciprocal translocations and the non-reciprocal translocations. So types of translocation: non-reciprocal intrachromosomal translocations, in which you can see the segment BC is translocated and it is within the same chromosome, that is intrachromosomal non-reciprocal translocation. And in this case you can see this is non-reciprocal interchromosomal translocation, in which case you can appreciate that the B and C is shifted from this chromosome to this over here, and this is the reciprocal interchromosomal translocation, in which AB segment is shifted over here and M and O segment is shifted over here. So the non-reciprocal interchromosomal translocation takes place between the two different chromosomes which come closer to each other during mitotic divisions. So reciprocal translocation yields either normal and can yield both the normal types and the defective gametes. So in this case you can say this is the A, B, C, D, E gene present on one chromosome and K, L, M, N gene present on another. And in this case you can see that the A, B, M, N and K, L, C, D. So M, N gene and C, D, E, they have been exchanged. These are the parent having the complete information. Now what will occur if these genes leads to the gametes, different gametes formation? So the normal gametes can be formed, that is A, B, C, D, E and K, L, M, N. In another case the A, B, M, N and K, L, C, D, E gene. So all the genes, all the complete information is also present, although there is a translocation, and this is the reciprocal translocation, but there is no genetic loss, so the gametes will be normal. In this case you can see there is the duplication of the AB gene in both the chromosome, and in this case you can see duplication of MN gene in both the chromosome with the loss of the CDE gene. So the deletion and duplication occur in these cases. So it means that the reciprocal translocation can yield to either normal or defective gametes. Types of translocations may also be Robertsonian translocations. In Robertsonian translocation, the centromeres from the two non-homologous chromosomes fuses and chromosomal material from short arm is lost. So 5% of the Down syndromic cases involve Robertsonian translocation between chromosome number 21 and chromosome number 15. So you can understand in this Robertsonian translocation, two non-homologous chromosomes lose their short arms and then they fuses at the centromeric position. So this chart shows you how different types of the gametes are produced if there is a Robertsonian translocation. So this one is normal. In this you can see two allelic forms of the chromosome number 14 and two allelic forms of the chromosome number 21 are present. But because of the Robertsonian translocation, the short arm of chromosome 21 and short arm of chromosome 14 is lost, and both the long arm fuses over here and they form a Robertsonian translocation. So when this cell leads to the gamete formation. So how many different types of the gametes are formed? The number one, which the cell which will having only the chromosome having Robertsonian translocation. The other type of the cell having both the 14 and 21 chromosomes which are normal. In third type, the chromosome number 21 is present normally and another is the chromosome which having the Robertsonian translocation. The fourth type, the only 14 chromosome is present. Fifth type, the 14 is present in addition to the one which is having the Robertsonian translocation, and in this case only the chromosome number 21 is present. So when all of these types of the gamete fuses with the normal gametes, what will be the result? So in this case, the normal pair of the chromosomes are added to this one. The patient will be monosomic, but as you can see, the long arm of the chromosome 14 as well as 21 both are present. So the patient will be a translocation carrier, although he may be monosomic having 45 number of the chromosomes. So first variant will result, that is the translocation carrier. In second variant, you can see all the normal are present. This is the diploid number of the chromosome and the patient will be normal phenotype. In third type, you can see then that the extra long arm of the chromosome number 21 is present in case of the Robertsonian translocation, although there will be diploid number of the chromosome, but because of extra copy of the chromosome 21 over here, the patient will show the translocation Down syndrome, which will be the third variant. And in fourth type, you can see chromosome 14 is normally present and there is the deficiency of the chromosome number 21. This is monosomy with 21 chromosomes. The total number of chromosomes will be 45 and it is lethal, not compatible with the life. Then another variant in which you can see the trisomy 21 is present, trisomy 14 is present, extra copy of chromosome number 14, and this is again lethal. And in this case you can see there is one deficient chromosome 14. This is monosomy 14 having total 45 number of chromosome, and this is lethal monosomy, not trisomy. This is lethal monosomy 14. It is not compatible with life. Third type of the structural abnormalities are inversions. So inversion, these inversions can either involve a portion of the centromere or it may leave the portion of the centromere. It will not include the centromere. So if it involves the centromere, these are called the pericentric inversions, including the centromere segment DEF is inverted in this chromosome, and if it do not include the centromere, the inversions are called the paracentric inversions. So segment BCD is inverted over here. The fourth type of anomalies or structural abnormalities are because of the duplication of the part of the genes on certain chromosome. So in this you can see the segment BC is duplicated over here in this chromosome. Here you can also see the BC is duplicated, and these are called the tandem duplicate. And if it is duplicated as well as inverted, these are called the reverse tandem duplications. And if they involve the end segment, the terminal segment, and there is the duplication, such duplications are called terminal tandem repeats. Another chromosomal abnormality is the uniparental disomy. In uniparental disomy, both copies of the chromosomes are inherited from a single parent due to error in the cell division. So examples are female affected with the rare X-linked disorders, sometimes Prader-Willi and Angelman syndrome. Fragile sites having more than 100 fragile sites have been identified in different chromosomes. So this is a chart which is showing the pattern of the uniparental disomy. These are the normal gamete having the haploid number of the chromosome, and after fertilization diploid number is restored. In this variant, this is the non-disjunction in both the parents. One parent having both the chromosome present and other parent having literally no chromosome. So all the genetic material is derived from the same parent, and it can lead to the uniparental disomy, one of the variant of uniparental disomy. In this you can see non-disjunction in one parent followed by duplication in other. This gamete is normal, and this gamete do not carry any genetic material. So when they fuses, they carry only half the amount of the genetic material, after which this chromosome duplicate itself, and so that all the material is derived from the same parent, and this is another variant of the uniparental disomy. In this you can see loss of the chromosome in zygote followed by the duplication. So zygote, loss of the chromosome in zygote, this chromosome is lost, and this then duplicate itself leads to the uniparental disomy. And in this variant you can see non-disjunction in one parent followed by loss in embryo. So this is diploid and this is normal haploid. This will initially lead to triploid, and then this one chromosome is lost. This chromosome is lost, and this leads to the uniparental disomy. Fragile sets more commonly found on the human X chromosome, and these are the different regions: the fragile site X, B site, C locus, fragile A and E and D and F locus. These are different loci which are present on the X chromosomes. Sex-linked dominant inheritance, the repeat sequence of the CGG variant, normal repeats occur nearly 5 to 50. There are 5 to 50 normal repeats of the CGG in the terminal region, while in prematuration phase there may be 50 to 200 repeats, and full maturation may be over 230 repeats are present. These are FMR1 gene and not expressing the promoter region that plays a role in synaptic connection of the nerve cells, not always expressed due to X inactivation. So you people know that this is the fragile X syndrome, and one of the X chromosome usually inactivated and it forms the bar body. So if that chromosome is affected, usually there are no symptoms. This is again fragile X syndrome is most common cause of the mental retardation. The distinctive physical appearance are the long face and the large ears are present. So the end notes are having wrong number of the chromosomes has serious, usually lethal consequences. Autosomal monosomies have even more catastrophic consequences than the trisomies, and the abnormalities must be consequences of an imbalance in level of the gene product encoded on different chromosomes. In trisomies make a smaller change than monosomy in those levels. And here I complete my today's class. This is the reference, that is Langman's Medical Embryology chapter 2. Thank you all students, dear students, for your attention for today's class, and Allah Hafiz till the next class.